Hello, welcome to World of Microbes. In the previous videos regarding blood we have discussed about plasma and erythrocytes in details. The link of the previous videos are in the description. In today's video we will discuss about leukocytes. We will also discuss characteristics, key functions, classification, life cycle and disorders of leukocytes. The leukocyte, commonly known as a white blood cell, or WBC, is a major component of the body's defenses against disease. WBCs are a cellular component of the blood that lacks hemoglobin, has a nucleus and is capable of motility. Leukocytes protect the body against invading microorganisms and body cells with mutated DNA, and they clean up debris. As part of your immune system, white blood cells circulate in your blood and respond to injury or illness. White blood cells protect your body against infection. As your white blood cells travel through your bloodstream and tissues, they locate the site of an infection and act as an army general to notify other white blood cells of their location to help defend your body from an attack of an unknown organism. Once your white blood cell army arrives, they fight the invader by producing antibody proteins to attach to the organism and destroy it. Your white blood cells are in your bloodstream and travel through blood vessel walls and tissues to locate the site of an infection. Contrary to their name, white blood cells are colorless but can appear as a very light purple to pink color when examined under a microscope and colored with dye. These extremely tiny cells have a round shape with a distinct center membrane, nucleus. You can only see white blood cells under a microscope, as they are extremely small. White blood cells account for 1% of your blood. There are more red blood cells in your body than white blood cells. White blood cell formation occurs in the soft tissue inside of your bones, bone marrow. Two types of white blood cells, lymphocytes, grow in the thymus gland, T cells, and lymph nodes and spleen, B cells. White blood cells originate from cells that morph into other cells in the body, stem cell, within the soft tissue of your bones, bone marrow. The production of leukocytes, known as leukopoiesis, is the biological process where the body generates new white blood cells to defend against infections and diseases. All leukocytes originate in the bone marrow from multipotent hematopoietic stem cells, hemocytoblasts, and develop based on chemical signals. Leukopoiesis divides into distinct pathways depending on the type of leukocyte needed. Myeloid lineage. This pathway produces cells that mature primarily within the bone marrow. It includes granulocytes, neutrophils, eosinophils, and basophils. They contain granules and primarily handle immediate innate immune responses, such as fighting parasites, bacteria, and allergens. Monocytes, are granular cells that enter the bloodstream and eventually migrate into tissues, where they differentiate into macrophages or dendritic cells to consume pathogens. Lymphoid lineage. This pathway produces lymphocytes, which include T cells, B cells, and natural killer, NK, cells. Lymphocyte precursors start in the bone marrow but complete their development and maturation in lymphatic organs like the thymus, lymph nodes, and spleen. They are responsible for adaptive immunity, producing antibodies, and regulating targeted long-term immune defenses. The production rate of leukocytes is strictly controlled by chemical messengers called cytokines, e.g., interleukins, and colony-stimulating factors. When the body detects an infection or inflammation, these signals stimulate the stem cells to increase the production of the specific leukocyte required to counter the threat. Leukocytes, white blood cells, are divided into two primary groups, granulocytes and agranulocytes, based on the presence or absence of visible granules in their cytoplasm. These cells act as the body's primary defense system to fight off infections, foreign invaders, and diseases. A breakdown of the differences, functions, and cell types reveals how each group operates. 1. Granulocytes, granular leukocytes. Granulocytes are characterized by a lobed nucleus and enzyme-filled granules in their cytoplasm that are released to destroy pathogens. They are primarily involved in the innate immune system, rapid, nonspecific defense. Neutrophils, basophils, and eosinophils are all granulocytes. These cells also all have azurophilic granules, lysosomes, and specific granules that contain substances unique to each cell's function. Histologically, granulocytes can be distinguished from one another by their nucleus's morphology, size, and how their granules stain. Agranulocytes, agranular leukocytes. Agranulocytes lack distinct, visible granules under a microscope and contain a non-lobed, mononuclear nucleus. They are central to the adaptive immune system, providing targeted, specific defense. Agranulocytes consist of lymphocytes and monocytes, and while they lack specific granules, they do contain azurophilic granules. Neutrophils are 12 to 15 m in diameter and have multi-lobed nuclei typically consisting of 3 to 5 segments joined by thin strands or isthmuses. Thus, they are also called polymorphonuclear neutrophils. Neutrophils contain specific granules in the cytoplasm that cannot be resolved by light microscopy, giving the cytoplasm a pale pink color. When activated, neutrophils migrate into the tissues via diapedesis. These cells have lifespans of a few days, and when activated in connective tissue, they undergo apoptosis and are then removed by macrophages. Eosinophils have a bilobed nucleus with large cytoplasmic-specific granules that are eosinophilic, staining red to pink. Basophils are 12 to 15 m in diameter, have bilobed or S-shaped nuclei, and contain cytoplasmic-specific granules, 0.5 m, in diameter that stain blue to purple. The basophilia of the granules is due to the presence of heparin and sulfated glycosaminoglycans. These cells have similar functions as mast cells. Monocytes are precursor cells for the mononuclear phagocytic system, which includes cells such as macrophages, osteoclasts, and microglial cells in connective tissue and organs. These cells constitute 4 to 8% of white blood cells, are 12 to 15 m in diameter, and have large nuclei that are indented or C-shaped, which can be eccentric. There is abundant cytoplasm, and the lysosomal granules at the resolution of the light microscope give the cytoplasm a bluish-gray color. Lymphocytes constitute approximately 25% of white blood cells, are of varying sizes, and have spherical nuclei. The small lymphocytes are similar in size to red blood cells, have spherical heterochromatic nuclei, and scant cytoplasm. Larger lymphocytes, such as activated lymphocytes, have indented nuclei and are 9 to 18 m in diameter with more cytoplasm containing azurophilic granules. Lymphocytes subdivide into several groups using the cluster of differentiation, CD, markers. The major groups are B lymphocytes and T lymphocytes. Neutrophils are the most common type of white blood cell in your body, acting as the primary first responders of your immune system to fight off infections and heal tissue damage. They make up roughly 40% to 70% of all circulating white blood cells. Produced constantly in your bone marrow, these highly mobile cells quickly travel through blood vessel walls to reach sites of acute inflammation or bacterial and fungal invasions. Neutrophils destroy pathogens using 3 main strategies. Phagocytosis, they physically engulf and swallow invading microorganisms. Degranulation, they release pre-stored microscopic packets of enzymes and toxic oxygen molecules to digest germs. Neutrophil extracellular traps, nets, they project stringy webs made of their own DNA to trap and immobilize pathogens too large to swallow. Doctors measure these levels through an absolute neutrophil count, ANC, during a routine complete blood count, CBC. As outlined by WebMD medical reference guides, a normal reference range typically falls between 1,500 and 7,700 neutrophils per microliter of blood. High neutrophils, neutrophilia count above 7,700 cells per microliter usually indicates that your body is actively reacting to something. Common causes include bacterial infections, physical injuries, intense emotional stress, smoking, or chronic inflammatory diseases. Low neutrophils, neutropenia count below 1,500 cells per microliter means your body has a weakened defense mechanism. This can happen due to viral infections, autoimmune conditions, bone marrow issues, or medical treatments like chemotherapy. Counts below 500 significantly elevate your risk for severe infections. Eosinophils are a specialized type of disease-fighting white blood cell, specifically a granulocyte, that plays a critical role in your immune system's defense against parasitic infections, allergens, and foreign organisms. They are produced in the bone marrow, make up a small percentage of your total white blood cells, and migrate into various tissues where they can survive for several weeks. Under a microscope, they are easily recognized by their distinct two-lobed, bilobed, nucleus and prominent cytoplasmic granules that stain bright brick red or pink when exposed to an acidic dye called eosin. Eosinophils act as major effector cells in human immunity, carrying out 2 primary jobs. Destroying pathogens, they target and kill large, multicellular foreign substances, particularly parasites and parasitic worms, like hookworms or tapeworms, by swarming them and releasing destructive enzymes directly from their granules. Driving inflammation, they release toxic proteins and chemicals that initiate and regulate inflammatory responses. While this helps trap and defeat localized infections, the same mechanism can cause tissue damage and trigger chronic symptoms in allergic disorders like asthma. Eosinophils are typically evaluated through a routine test called a complete blood count, CBC, with differential percentage. They generally account for 1% to 4%, or less than 5%, of all circulating white blood cells. Absolute count, a healthy absolute eosinophil count, AEC, typically ranges from 0 to 500 cells per microliter of blood. An abnormal level of eosinophils can point to specific underlying shifts in your health. Eosinophilia, when your body produces an unusually high volume of these cells, exceeding 500 cells/microliter, it is known as eosinophilia. This condition is most commonly triggered by allergies, asthma, drug reactions, eczema, or parasitic infections. In rare cases, extremely elevated counts, hypereosinophilia, can cause the cells to exit the bloodstream and pool inside vital organs like the lungs, heart, or gastrointestinal tract, causing localized tissue damage and organ inflammation. Low counts, eosinopenia. Having a very low or virtually non-existent eosinophil count is generally not a cause for medical concern because other components of your immune system easily compensate for them. Low levels are typically discovered by chance and are often caused by the use of corticosteroid medications, acute severe infections like sepsis, or Cushing syndrome. Basophils are a rare type of white blood cell produced in the bone marrow that play a crucial role in your immune system's response to allergens, pathogens, and parasitic infections. Although they are the largest type of granulocyte by size, they are the least common, typically accounting for less than 1% of your total circulating white blood cells. Basophils act like a security alarm system for your body. When they encounter a foreign invader or an allergen, they quickly open up and release specialized chemical messengers stored in their internal granules. Histamine, this vasodilator, widens blood vessels and increases blood flow to damaged tissue. While this helps your immune system deploy resources, it also triggers classic allergy symptoms like swelling, itching, hives, and runny noses. Heparin, a natural anticoagulant that thins the blood and prevents unwanted blood clots from forming too quickly, keeping blood flowing smoothly through inflamed areas. Cytokines and chemokines, these signaling molecules, like interleukin-4, recruit other immune cells, such as eosinophils and T cells, to help fight off the perceived threat. When clinicians look at a blood smear under a microscope using a basic purple dye, like the Reichgem stain, basophils are easily identified by distinct features. Dark granules, they are packed with large, coarse, blue-black or deep purple granules that often completely crowd out and obscure the nucleus. Multilobed nucleus, they typically feature an irregularly shaped nucleus containing 2 to 3 lobes, often resembling a C or S shape. Size, they average roughly 10 to 15 micrometers in diameter. A standard complete blood count, CBC, with differential test measures your basophil levels. The typical normal reference range is very low, 0 to 100 cells per microliter of blood, or 0% to 1% of total white blood cells. When your absolute basophil count is elevated, it means your immune system is actively fighting something or experiencing chronic stress. Common causes include allergic reactions, chronic inflammation, infections, hypothyroidism, myeloproliferative disorders, low basophils, basopenia, severe, acute infections that deplete your cell supply, overactive thyroid, hyperthyroidism, severe ongoing stress or the prolonged use of corticosteroid medications. Monocytes are the largest type of white blood cell in the immune system, acting as the body's primary cleanup crew to destroy pathogens and remove dead tissue. They are produced in the bone marrow, travel through the bloodstream for a short time, and then migrate into various tissues where they mature into specialized defenders called macrophages and dendritic cells. Main functions include phagocytosis, engulfing and digesting cellular debris, harmful bacteria, and viruses. Antigen presentation, displaying parts of destroyed germs to T cells, triggering a targeted immune response. Cytokine production, releasing chemical signals that regulate inflammation and coordinate other immune cells. Monocyte levels are measured using a complete blood count, CBC, with differential. Healthcare providers is typically 2% to 8% of your total white blood cells. High levels, monocytosis. This usually means the immune system is actively fighting something. Common causes include chronic infections, like tuberculosis, autoimmune disorders, like lupus or IBD, physical stress, or recovery from an acute infection. In rare cases, persistent high levels point to certain blood cancers like leukemia. Low levels, monocytopenia. 0.2 times 10/L. This occurs when the body's bone marrow production is suppressed. Common causes include chemotherapy, radiation therapy, bone marrow disorders, or severe bloodstream infections like sepsis. Macrophages are a specialized type of large white blood cell belonging to the innate immune system. Derived from the Greek phrase for big eaters, their primary function is phagocytosis, a process where they engulf, digest, and destroy pathogens, cellular debris, and cancer cells. They act as both the frontline security guards and the central housekeeping crew for every organ in the human body. Phagocytosis, they wrap their cellular membrane around invaders like bacteria and viruses, pulling them into an internal pocket called a phagosome. This fuses with a lysosome filled with harsh enzymes to dissolve the threat. Antigen presentation, after digesting a pathogen, they display pieces of its proteins, antigens, on their surface. This alerts and activates adaptive immune cells like T cells and B cells. Tissue repair and regeneration, they clear away dead cells and secrete growth factors that encourage new blood vessels and tissue patches to form. Homeostasis maintenance, they recycle old red blood cells in the spleen and capture iron to keep the body's baseline balanced. While some macrophages wander freely through tissues, many settle down long-term into specific organs where they assume unique names and highly tailored housekeeping roles. Microglia, found in the central nervous system, they protect neurons and trim away redundant neural pathways. Kupffer cells, residing in the liver, they filter out gut-derived toxins and recycle old blood elements. Alveolar macrophages, stationed inside lung air sacs, they neutralize inhaled dust, smoke particles, and microbes. Osteoclasts, located in the bones, they continuously break down and reshape bone tissue to maintain mineral levels. Most tissue-resident macrophages are deposited during embryonic development and maintain themselves through local cell division. However, during an active infection or injury, the bone marrow ramps up production of monocytes. These immature cells travel through the bloodstream and cross into damaged tissue, where they quickly expand and mature into fully functioning macrophages. When working properly, macrophages keep tissues clean and free of disease. However, if their signaling pathways are subverted, they can drive chronic disease. For instance, chronic M1 activity fuels autoimmune disorders and cardiovascular plaque buildup. Conversely, tumors can trick M2-like macrophages into shielding cancer cells from the immune system, inadvertently promoting metastasis. Dendritic cells, DCs, are specialized, bone marrow-derived white blood cells that act as the master messengers of the immune system by linking innate, fast, nonspecific, and adaptive, slow, highly specific, immune responses. They are named for their unique, tree-like structural projections called dendrites, which provide a vast surface area to capture invading pathogens, cellular debris, and toxins. Discovered in 1973 by scientists Ralph Steinman and Zanvil Cohn, they are classified as the most potent professional antigen-presenting cells, APCs, in the human body. The immune sentinel life cycle. Dendritic cells operate in a highly synchronized two-stage life cycle. The immature, surveillance, stage. In their immature state, DCs patrol the body's environmental frontiers, such as the skin, lungs, and gut lining. They are highly efficient at eating or drinking substances from their surroundings through phagocytosis and macropinocytosis. Using specialized pattern recognition receptors, PRRs, they screen the environment for molecular damage or pathogens. The mature, presentation, stage, once a dendritic cell intercepts a foreign threat, antigen, it digests the invader into tiny protein fragments. This encounter triggers a rapid maturation process. The DC leaves its resident tissue, travels through the lymphatic system, and settles into local lymph nodes. Here, it presents the foreign protein fragments to naive T cells via major histocompatibility complex, MHC, molecules, effectively teaching and activating the T cells to launch a targeted attack. Major subsets of dendritic cells. Dendritic cells do not belong to a single, identical population, they exist in distinct subsets tailored for specific defensive tasks. Conventional/classical DCs, CDCs, the main drivers of standard T cell activation. They excel at sensing physical tissue injuries, processing standard microbial threats, and presenting foreign proteins to both CD4+ helper and CD8+ cytotoxic T cells. Plasmacytoid DCs, PDCs. These cells possess a shape resembling plasma cells and serve as the body's primary antiviral defense units. Upon encountering a virus, they rapidly secrete massive amounts of type 1 interferons, IFN-alpha, which block viral replication and sound an alarm to neighboring cells. Langerhans cells, LCs, a unique subset of dendritic cells localized strictly within the epidermal layer of the skin. They act as the very first line of cellular immune defense against cuts, insect bites, and superficial infections. Monocyte-derived DCs, MODCs. Unlike homeostatic DCs that develop continuously from stem cell progenitors, MODCs are emergency cells. They differentiate rapidly from inflammatory monocytes in the bloodstream only during active, severe bouts of tissue infection or inflammation. Lymphocytes are a specialized type of white blood cell that serve as the primary cells of the adaptive immune system. They originate in the bone marrow, circulate through the bloodstream and lymph vessels, and concentrate heavily in lymphoid organs like the spleen and lymph nodes. They make up roughly 20% to 40% of all circulating white blood cells. Three primary types of lymphocytes. B cells, B lymphocytes. These cells mature in the bone marrow. They produce customized proteins called antibodies to neutralize target pathogens. They also generate long-term immunologic memory. T cells, T lymphocytes. These cells mature in the thymus. They handle cell-mediated immunity by destroying infected or cancerous cells directly. They also coordinate the broader immune response. Natural killer, NK, cells. These cells belong to the innate immune system. They rapidly identify and kill virus-altered cells or tumors without needing prior exposure. Although leukocytes and erythrocytes both originate from hematopoietic stem cells in the bone marrow, they are very different from each other in many significant ways. For instance, leukocytes are far less numerous than erythrocytes. Typically there are only 5,000 to 10,000 per L. They are also larger than erythrocytes and are the only formed elements that are complete cells, possessing a nucleus and organelles. And although there is just one type of erythrocyte, there are many types of leukocytes. Most of these types have a much shorter lifespan than that of erythrocytes, some as short as a few hours or even a few minutes in the case of acute infection. One of the most distinctive characteristics of leukocytes is their movement. Whereas erythrocytes spend their days circulating within the blood vessels, leukocytes routinely leave the bloodstream to perform their defensive functions in the body's tissues. For leukocytes, the vascular network is simply a highway they travel and soon exit to reach their true destination. When they arrive, they are often given distinct names, such as macrophage or microglia, depending on their function. As shown in Figure 1, they leave the capillaries, the smallest blood vessels, or other small vessels through a process known as emigration, from the Latin for removal, or diapedesis, dia equals through, pedin equals to leap, in which they squeeze through adjacent cells in a blood vessel wall. Once they have exited the capillaries, some leukocytes will take up fixed positions in lymphatic tissue, bone marrow, the spleen, the thymus, or other organs. Others will move about through the tissue spaces very much like amoebas, continuously extending their plasma membranes, sometimes wandering freely, and sometimes moving toward the direction in which they are drawn by chemical signals. This attracting of leukocytes occurs because of positive chemotaxis, literally, movement in response to chemicals, a phenomenon in which injured or infected cells and nearby leukocytes emit the equivalent of a chemical 911 call, attracting more leukocytes to the site. In clinical medicine, the differential counts of the types and percentages of leukocytes present are often key indicators in making a diagnosis and selecting a treatment. If you have a low white blood cell count, you are likely to get infections, leukopenia. If your white blood cell count is too high, leukocytosis, you may have an infection or an underlying medical condition like leukemia, lymphoma, or an immune disorder. Symptoms of white blood cell conditions, where you may have a count that is too high or too low, include fever, body aches and chills, wound that is red, swollen, oozes pus or won't heal, frequent infections, persistent cough or difficulty breathing. A complete blood count, CBC, test identifies information about the cells in your blood. A lab completes this test after a medical professional draws your blood and examines your white and red blood cell count. White blood cells scan is a test to detect infection or abscesses in your body's soft tissues. This test involves withdrawing your blood, separating the white blood cells from the sample, tagging them with a radioactive isotope, returning those white blood cells back into your body. Then an imaging test will identify areas that show infection or abscess on your body. T is normal for you to produce nearly 100 billion white blood cells each day. After completing a blood draw, a test counts your white blood cells, which equals number of cells per microliter of blood. The normal white blood cell count ranges between 4,000 and 11,000 cells per microliter. Causes of low white blood cell count include bone marrow failure, aplastic anemia, bone marrow attacked by cancer cells, leukemia, drug exposure, chemotherapy, vitamin deficiency, B12, HIV, AIDS. A blood test with fewer than 4,000 cells per microliter of blood diagnoses low white blood cells. Causes of high white blood cell count include autoimmune disorders, lupus, rheumatoid arthritis, viral infections, mononucleosis, bacterial infections, sepsis, physical injury or stress, leukemia or Hodgkin's disease, allergies. A blood test with more than 11,000 cells per microliter of blood diagnoses high white blood cells. Treatment for white blood cell disorders vary based on the diagnosis and severity of the condition. Treatment ranges from taking vitamins, taking antibiotics, surgery to replace or repair bone marrow, blood transfusion, stem cell transplant.